start this. And Dr. Wu, I don't know, are you able to share your screen? If you have anything you want to present, please feel free.
Okay, let me try to upload my presentation.
Okay, thank you.
Okay, all right. So I guess you guys can see the presentation now.
Yes, yes.
Okay. Well, first I would like to thank Scott and everybody for inviting me to this event. And I have always been interested in education outreach within NASA, and I used to serve as both the chair as well as the member of the technical committee for some of the NASA centers. And as a matter of fact, I worked with Misty Moore very closely previously. And if Brandon or Scott sees her, please say hello. And when I received the original request to give a presentation, I think the request came from Dr. Taraboletti, because the request was to discuss about metabolic information used for radiation by dosimeter. And then I said, well, I haven't done anything in that area, but then of course, we have done a lot of work in biological dosimetry for astronauts, so not necessarily using metabolic activity information, but they're using chromosome damage data that we collected from the astronauts. So this presentation actually was a little bit more focused, perhaps, than what the intent was for this exchange event. But regardless, I'm just going to discuss first what space radiation is, and then discuss the biodosimetry results that we published early this year. And when we have the exchange with faculty members later, and I can talk about some of the other areas that would certainly be of interest to some of the faculty members here. For instance, microbial research, and that's one of the areas. And I see a lot of people from the chemistry department, and yes, water quality, and that's one of the air quality and water quality, of course, are the one of the major concerns, environmental concerns for astronauts in space. I think for those who are in the chemistry departments, I'm sure you can find some common interest in there. So I'm just going to start by introducing the space radiation environment, because that's my area. And I believe that's what Dr. Taraboletti's area is at now. And so space radiation, well, I may get into a little bit of the basics here, and that is space radiation is somewhat different from gamma rays or X-rays that we are accustomed to, on the ground, and because that's mostly photons. But then space radiation will be charged particles. Just to think of a cell nucleus, and here, and this is an atom with all the electrons stripped away. Then you have a cell nucleus, which would consist of protons or neutrons. And so this will carry a positive charge and moving at the extremely high speed. And that's what cosmic radiation consists of mostly. And then there are several different sources of cosmic radiation, and depending on where you are in space. In the low Earth orbit, or the orbit of the space station or space shuttle previously, and the astronauts are exposed to mostly protons trapped in the geomagnetic field. So what you see here is that you have a geomagnetic field line going from the North Pole to South Pole, and you see that because of the asymmetry between the rotation axis and the geomagnetic field, and you see a asymmetry of the trapped radiation belt. And what you find here is that in the altitude of several hundred kilometers, and only when the spacecraft passes through this particular region, which is called South Atlantic anomaly, the astronauts will be exposed to trapped protons. And for engineers, I mean, this is not strange, because this is just the energy spectrum of those particles, and you can see that most of the particles can reach energy as high as several hundred MeV, or million electron volts. And for those low energy ones, although they are abundant, but they can be stopped even somewhat by your clothes, or certainly by the shield of the spacecraft. So that would be the first source. And then the second source is the galactic cosmic radiation, which is present not only in the low Earth orbit, but also in free space. And GCR now consists not only of protons, but then a whole range of different elements of particles. And as you see here, and this is the relative abundance. And now the energy spectrum, you can see that the sum of those particles can reach the energy as high as 1,000 or several thousand MeV per nucleons. So those would be considered very high energy. And then the third source will be the particles released from the solar particle events from the sun. And so those are mostly protons, a little bit of helium. And so when those particles release from the sun, and then they can reach the Earth and penetrate into the lower atmosphere, and producing the northern lights or southern lights effect. And although we can't quite predict when the sun will release those particles, but then in general, the solar particle during the solar activity period, you have more chance to have those solar particle event in comparison to solar minimum activities, as you see here. And I do want to mention here also, and with the trapped protons, or GCR, the exposure scenario will be chronic. Chronic meaning that the astronauts will receive a certain amount of a dose over a period of a year or 2 years. And whereas, I mean, for large solar particle events, the astronauts can receive a high enough dose to experience those what we call acute radiation syndrome. So this is kind of just a summary of the space radiation environment. Again, I mean, you have a trapped radiation, and when you travel outside of the Earth and to the Moon or Mars, and then the astronauts will be exposed to GCR, or solar particle protons. And here basically shows the map of the Earth. I mean, if you look at the background, which is the map of the Earth, and the US is here, and South America, Africa is here. And this is where the South Atlantic anomaly is at. So it's when the spacecraft passes through this region, and the astronauts will be exposed to the trapped protons. And you see that there's a cutoff of 57 degrees here, because this map is mapped by the I think Russian Mir station, which only moved from the inclination of a minus 57 degrees to a positive 57 degrees. All right, so that kind of summarizes the space radiation environment. But then the question is, well, how much radiation they receive during space activities? And of course, I mean, it will all depends on many different factors. The duration of the mission definitely will be one of the major factors, and the inclination and the altitude also affect the dose that the astronauts receive. And I show here a couple of examples of space shuttle missions and STS-94 and 95. And you can see that during the STS-94 activities, the altitude is about half of STS-95. And 95 being so high because this was one of the missions for Hubble telescope repair. And they all have the same inclination of 28.5 degrees. And basically, this is the inclination when you launch from Kennedy Space Center and straight toward the east, and that's the inclination of the orbit that will achieve. But then if you were to launch the spacecraft from Russia, and that's when you get the 57 degrees. And now you see that the duration of one is about twice as high as this. But now I'm not sure you can see it on my screen. It gets covered. The dose actually is about 10 times less. I mean, in STS-94 in comparison to STS-95. All right, and of course, I mean, this shows that the dose received by the astronauts during different missions, Mercury missions sometimes last only a few hours, and now with the International Space Station missions that last for about 6 months, the astronauts will receive a dose about 10 centisieverts. And how would that 10 centisievert compare to the other exposures we receive in our daily life? And I just mentioned here that the US limit for occupational exposure to radiation is 5 centisievert per year, and that's both the OSHA's limit as well as DOE. So you can see that immediately that for astronauts, the exposure over a period of 6 months in space have already exceeded the radiation limit set for radiation workers on the ground. So now, with the space radiation, then of course, I mean, what we are concerned about for astronauts' health. Well, within NASA, there is a human research program which has identified a number of risks for astronauts. Then the first of which, of course, as you would have guessed, is cancer, and cancer risk is one of the major ones, but then it's not the only one. And central nervous system damage, and that's another concern. So basically what happens is that when the space radiation or those high energy particles penetrate through the spacecraft and through the brain, and we want to make sure that it will not damage the CNS, and that will affect both the early effect that will affect the success of the missions. For instance, they have to be able to land the spacecraft on the moon or Mars. And some of the later CNS risks will include, for instance, Alzheimer's disease and other brain-related diseases. Degenerative tissue damage that would include, for instance, cardiovascular diseases or a cataract that we have already observed in the astronauts. And acute radiation syndrome, that would be experienced. Oh, gee, I don't know what happened to my presentation. Acute radiation syndrome will be experienced during the solar particle event, and those symptoms will include, for instance, vomiting, nausea, and then blood count reduction. Okay, so NASA Human Research Program supports this facility at the Brookhaven National Lab. So mainly what we do is that we use one of the accelerators, and actually this is a feeder accelerator for the bigger and higher energy accelerator here. And so this will generate those particles from protons to as heavy as gold ions at very high energy. So basically, NASA will support those investigators to conduct experiments at Brookhaven by exposing both cells or mostly animals, rodents, to those particles to study different effects that will relate to those risks that I just discussed. All right, and so that's of course, we have identified a number of risks, but then what have we seen? I mean, what kind of biological effects we have seen in astronauts from exposure to space radiation? Well, actually, there's a number of them. We probably can't say a whole lot of about cancer. I mean, for one, is that the number of astronauts is still low, so we probably don't have sufficient statistics. And for two, is that it's difficult to find the comparison groups for the astronauts to compare to, because after all, they are super healthy individuals. But there are a number of effects that have been reported in astronauts that's due to exposure to space radiation. And the first of which it will be light flash. And light flash was experienced first by the Apollo astronauts when they flew to the moon. So what happened was that when they tried to close their eyes and they start to see those flashes, and sometimes it can be a dot or maybe a couple of dots and maybe some cloud looking effects. And so what happened is that while our eyelid can stop the penetration of photons, but it cannot stop the high energy protons or other particles. And when they stimulate the retina, then they could produce this type of effect that has been reported in the astronauts. Then the second effect will be cataracts. Cataracts that are known, and so this is a work from one of my colleagues, Frank Cucinata. So what Frank found was that if he divided the astronauts into two groups, one group will receive a higher doses of radiation in the eye, and the other group will receive lower doses of radiation in the eye. And then what he found that eventually everybody, just like us, everybody will develop cataract at some age. But then the group of astronauts that receive a higher dose of radiation in the eye will develop cataract at a younger age. And then the third effect is what we do in my lab now, and that's the chromosome aberrations observed in the astronauts' blood cells. So what happened is that so this is the DNA you have in the cells. Once DNA condenses into chromosomes during mitosis, and if the DNA or chromosomes are damaged, and then there's a rearrangement of those damages can be detected under the microscope with some of the techniques. And in this case, the technique we use is called fluorescence in situ hybridization, or FISH. And we, with the technique, we can paint, quote unquote, paint different chromosomes in different colors. And for instance, now this red chromosome now is broken into several different pieces and has translocated into other chromosomes. So you can use this technique to detect the chromosome damage or the DNA damage in the astronauts' blood cells. And this is what we have found. And we did that type of studies in relationship to what we call biodosimetry, and that kind of related to the request that I received. And biodosimetry meaning that we can use some biological markers in the body to determine how much exposure that the individual will receive. And this has been an active research area within the Homeland Security or Department of Defense, because if terrorists, for instance, drop a dirty bomb in New York, and we need to know how much exposure those individuals will receive in order to administer proper treatment for those victims. So that's the reason. But then for us, for astronauts in space, and they do wear a radiation badges, and which is different from the general population. But then with a radiation badge, which will detect how much radiation they receive in space activities, then why do we still need to perform a biodosimetry? And actually, there are a number of reasons, and I list right here. And of course, I mean, it will complement the measurement using physical dosimeters, but then most importantly, and the biological damage we detect in the human body, or in this case, the astronaut's body, will take into account of the individual susceptibility, will take into account of the self-shielding effect of the body, and perhaps take into account of the potential synergistic effect with other space flight factors such as microgravity. And hopefully we can use this some biomarkers to predict the risks. Right, then there are many different methods people have proposed for biodosimetry over the years. And I have to say that I am not familiar with metabolism, and I'm going to mention about how metabolism can be related to some of our risks. But then for radiation detection, yes, I mean, radiation will produce many different events. Definitely will affect metabolic activities, will affect some of the immune dysfunction, for instance, will be caused by radiation exposure. But I think, but among many of the different suggestions that we have reviewed, actually we decided to use what we call the gold standard, which is the chromosome damage. And other methods, of course, have been looked at by other investigators, mostly, I mean, by using the expression of DNA damage response genes. And in this case, I just show this as an example and of the study we did, and in which case we exposed human blood cells to radiation and investigated the dysregulation of some of the DNA damage response genes. And we found that, of course, some of the genes, FD, XRRBBC3, are upregulated, other genes might be downregulated. And actually, we performed this analysis and transcriptomic analysis or gene expressions in all of the genes. And we found that some of the gene expression changes perhaps only occur in certain subtype of cells, in this case CD4 cells, but not in other subtypes like the CD8 cells, for instance. However, I can tell you that I also have another NASA project, which is to investigate transcriptomic changes in the ISS astronauts' blood samples. And we are in the process of analyzing those data. However, from the data we have seen, perhaps we don't see any of those DNA damage response genes being upregulated or downregulated. And what we have seen are mostly just the genes related to stress. So it's mainly just the stress response genes. And this stress can be caused by microgravity, can be caused by radiation, but we actually don't know which environmental factors is the major contributor. But then my personal opinion is that most of the stress will be caused by microgravity rather than by space radiation. Okay, then getting back to the chromosome, using chromosome method for performing biodosimetry analysis or cytogenic biodosimetry. So what we did was that then we collected blood from the astronauts before the flight and after the flight. And once the blood samples were collected, and then we would culture the blood cells, and we stimulate the cells to grow. And by the time they reach mitosis, we process the chromosomes and we painted those chromosomes in different colors, and we can visualize those damage under the microscope. And this is so the remaining of the presentation. I'm just going to briefly go through some of the results in our biodosimetry study. And so far, we have collected samples from 43 ISS crew members, and for the samples or blood samples collected pre-flight, and we also exposed those blood to gamma rays at ex vivo, so not in the body, but outside of the body, to a set of doses between 0 and 2 grays. And then the post-flight samples, and we collected at 2 different times. And then the first time point, it was within 2 weeks after landing. And then the second time point is about 6 to 12 months after landing. And that's during their next physical at Johnson Space Center. And 5 of those 43 crew members flew on the ISS twice. And I said that also earlier that we use this fish painting technique to analyze chromosome damage. So this is kind of summarizes the results. So what you see here in this box is that this is a chromosome aberration frequencies. Okay, just don't pay too much about attention to the unit or everything, but basically what you see is here is that this is a pre-flight chromosome aberration rates in the astronaut's body, and they vary quite a bit among the individuals. And this would be the same if I were to draw blood from the 10, 15 people in the audience, and the background of chromosome damage in our blood samples will vary considerably as well. And this is plotted as a function of the dose the astronauts received in flight and measured by the physical dosimeter. And you can see that after flight, and then the damage now as a whole now have increased significantly from the pre-flight level. And the dose they receive now is generally it's about 0.02 or 0.03 gray. Now at the bottom again, I mean this is a pre-flight, and then this is a post-flight, and this will be the dose response or the chromosome damage in the blood samples after exposure to gamma rays outside of the body. And so you see a clear dose response here. And what this dose response tells us is what we call radiosensitivity. So now this is an example of the dose response from 9 individuals. And that tells us the radiosensitivity of individuals. And what you see here, this radiosensitivity for this particular individual, for instance, individual 9 will be have a higher slope. So this particular individual will be somewhat higher, have a higher radiosensitivity than, for instance, in comparison to subject number 7 in this case. So now we perform some of the analysis. Basically, we want to answer this question, and that is whether the radiosensitivity we determined from the pre-flight blood samples will predict or their damage that's caused by space radiation during flight. So the analysis is through some Poisson regression. Actually, this was done by my biostatistician colleagues, so I won't get into the details. And I'm just going to go through some of the results quickly. And this is by comparing the background, okay, chromosome damage. This is y-axis. That is the background chromosome damage among the individual crew members, and plotted as a function of the average slope. And just look at the slope as the radiosensitivity now. So what you see here is very interesting, and that is, I don't know what happened. What you see here is interesting because there's a positive correlation between the background and the slope. So which means that this data was collected from the individuals even before they flew in space. So what this shows is that for individuals that have a higher background or have a higher radio sensitivity will have a background. And then you can think that for most of us, then what causes the damage in their blood cells, and that will be from perhaps from smoking or from exposure to the toxic chemicals or from exposure to x-rays during medical procedures such as x-rays or CT scans. But then, so what this shows is that there's a positive correlation, and which means that perhaps the individuals with a higher background and or with a higher radio sensitivity will be also sensitive to to other environmental stressors. Another interesting findings from this analysis is the age effect. The age effect is that so here we the top figure showing the background chromosome damages among those individuals. I cannot plot the scattered plot here to show the individual age of the crew members, because otherwise you'll be able to identify them. So that's the reason I group into minus 45 years old and between 45 and 50, and some of them are above 50 years old. So what you can see here is that there's a clear age dependence, meaning that the older individuals will have more chromosome damage in their body, which actually is not new. And this actually was from a paper by Jim Tucker and published a long time ago, 1994, in which he analyzed chromosome damages among the individuals of different age groups, and have found that yes, I mean the older people will have more damage than the younger people, and this is not unexpected. So what's unexpected here is that the radio sensitivity and also increases as a function of age, meaning that the older individuals are more sensitive to radiation damage than younger individuals. And this figure kind of answers our question. Yes, the pre-flight average slope, or this is radio sensitivity, does correlate with the post-flight chromosome damage that we observe in the astronauts. And in addition, I mean, we looked at those 5 crew members who flew on the ISS twice. And of course, I mean, then the question we try to address is that this prior mission on the ISS will affect their radio sensitivity or not. And to answer this question, so we compared the results from both flights, and we found that as far as the slope or radio sensitivities, perhaps statistically there's no difference between the two. But then if you look at the baseline, of course, after the first flight, then the astronauts will have a higher baseline. So the baseline chromosome damage will be higher for the second flight in comparison to the first flight, and that's also expected. And I won't get into this RBE because that kind of requires some explanation. Yes, and then this is the last slide. And so what we also have done is to compare those damage between the samples collected 2 weeks after the mission and the sample collected 6 months or 1 year after landing, and we didn't find a significant difference between these 2 time points. And this indicates that the damage in the body is not damage in the circulating blood cells, rather it's a damage in the bone marrow cells, and that would those damaged bone marrow cells would then generate those cells that we detect. So I won't read this, but basically this kind of summarizes some of the results that we collected from this biodosimetry study. And also like to thank my colleagues for this particular study. Right, I think that's all I have. So I hope that this gives you some idea about the space radiation environment and about some of the concerns we have. And so with the radiation, with a couple areas, perhaps, I mean, you can contribute to. And of course, I mean, contribution in the development of dosimetry or biodosimetry. And that's perhaps related to some of the research areas in some of the faculty members in the chemistry department, and that's to develop sensitive radiation dosimeters and perhaps using some nanomaterials. And because I know that I support some of the studies even within NASA in that area. I mean, whether you can, so that's a physical dosimeter using nanomaterials. If you can develop something like that, and that will be relevant to NASA, particularly, I mean, if you can develop some dosimeters that can detect neutrons. And I didn't mention about the neutrons earlier, and outside of the spacecraft, there will be mostly charged particles. But there, as those particles penetrate through the spacecraft or hitting the moon or Mars, they generate secondary neutrons. And detection of neutrons, of course, has been a challenge. And biological dosimeters, of course, any sensitive methods that will be more sensitive than chromosome, and certainly, I mean, that will be of interest. And but then I said that I don't know how much Dr. Tarabaletti's study can be applied to the development of biodosimeter, but for space radiation exposure and for perhaps for homeland security, that's applicable. But then for space radiation exposure, the dose is low and then the exposure is chronic. So we really need to do a lot of studies to determine whether any of those information can be used. But metabolism in space definitely is a question that we try to address. And metabolism that's related, of course, to both nutrition intake and also to drug efficacy in space. So those are certainly some of the key questions we try to address. And other areas, I think somebody was, I think somebody is studying microbial and microorganisms in space, of course, will be related to both astronauts' health, because we need to control the microbial growth in space. So actually within my branch, there's a microbiology lab which addresses those questions. Microorganisms, growth, survival in space, and that's another fundamental space biology questions that NASA asks. And I try to think of other areas. Perhaps, I mean, I will listen to the presentations by other faculty members. Perhaps I will think of something else. Okay, I think that's all I have now.
Great. Thank you, Dr. Wu, for this comprehensive presentation. I guess before we move on to our faculty presentation, if any immediate questions you want to ask, maybe we can open it up for questions for a few short questions at this point. So please go ahead. Yeah, if anyone has any question you want to ask, please feel free. Okay. Well, if there's no question at this point, maybe we can just move on to the presentation from UEDC side. I don't, I would, suggest maybe whoever you want to present, just feel free to, share your screen and the slides, and I think, let me see here. Yeah, we'll have a couple of faculty interested in presenting today. So, so please feel free to share your screen, and then we can listen to your research. Anyone want to go first? Please feel free.
Dr. Zhu, I'll follow up since I'm kind of going to be on a similar talk as Dr. Wu just went. So I'll share my screen. Give me 1 second.
Okay, go ahead.
Can you see my screen okay?
Yes.
All right, excellent. So I don't want to take away from our other professors, so I'm going to quickly go through a bulk of my research, and again, hopefully see some overlap with NASA. I've worked generally in metabolomics and understanding disease, injury, and then also radiation exposure. So just to clarify quickly, metabolomics is this field of an omics field where we're characterizing small molecules or metabolites in cells, tissues, and organisms. And typically, I do that using mass spectrometry, either through what we call untargeted profiles, where we're just kind of collecting everything and then looking at pathway analysis and getting an understanding of maybe what's changing comparatively. Or you could do this targetedly, where you are looking at molecules of interest, quantifying them, and validating kind of what's happening. Again, my research I wanted to share with you have looked at using metabolomics in disease, particularly neurodegeneration and demyelination, and also in injury and environmental exposure. So I've looked at oxidative damage, radiation damage, and also I've looked at drug protection for those environmental exposures. In particular, I've used a model of oligodendrocytes, which you can see right here on my screen is the cell. It actually is a type of glial or brain cell that supports neurons and also helps with astrocytes. So this is one of many cells besides the neuron that's actively involved in our metabolic support of the neurons in the brain. So because these are a highly metabolic cell, I'm interested in how these are reacting in the brain because of their tie to metabolism. So I've done some investigation in neurodegeneration, specifically looking at models, actually of viral models. So there's a lot of viral mouse models that cause nerve inflammation or demyelination. And also there is one particular model that is a model using a chemical, small molecule chemical called the Cooperzone model. It was really interesting because it's just one small single molecule. Here it is not bound to copper, here it is bound to copper. And yet when it's fed to mouse, it causes the death of those oligodendrocytes and then causes demyelination in the brain. So we did an investigation using both cells and mice, looking at what happens metabolically to those oligodendrocyte cells and also what happens to the brain tissue itself. Again, using metabolomics, and we were able to identify some particular metabolites that were related to this neurodegeneration. Generally, what we were able to discover is that cuprazone, this small molecule, was reacting with a vitamin precursor, pyridoxal 5-phosphate, and we showed that it actually could bind using mass spectrometry and thus cause other downstream metabolism effects in the oligodendrocytes. The other area, and the one I really wanted to focus on today because of the shared work, is my work in radiation metabolomics. Radiation metabolomics that I used it in was used as a means of biodosimetry, which we've just had a really nice review on what biodosimetry is. So using biological markers, in this case metabolites, to be able to identify exposure of different sources of radiation, different levels, and different time intervals. And we did this using untargeted metabolomics. So I've worked in many different biofluids, urine, blood, etc., and used those to try to see patterns in biodosimetry. One area of interest that I worked in was actually testing out a small polymeric device that collected blood. It was able to store and collect blood without using a freezer, and then being able to then extract that blood and test it for differences in radiation exposure. And you can see this is a cellulose-based polymer that we used, and it was the best one that we found. We were able to identify distinct metabolite differences between our non-irradiated or sham and 8-gray irradiation. This is from mice, by the way. And again, using this pattern of metabolites, we could develop a feature panel that could allow us to distinguish. This was kind of a test of the protocol and being able to use that particular device. We also have looked at feces and paired metabolomics with kind of the microbiome, where we did metagenomics as well. And this trial was actually a long duration trial using gamma radiation in non-human primates, looking at pre and post time periods and seeing this dysregulation of metabolites. So we saw this kind of heightened at a particular day, day 3, in their feces as far as dysregulation of metabolites. And we were able to pair this as well, like I said, with metagenomics to see how the gut microbiome was affected. This one was really interesting because we saw a lot of pathways associated with hormones, hormone production through the actual microbiome as well, bile acids, not surprising, and some other inflammatory pathways associated with vitamin D. And again, here's some of those metabolomic profiles we saw over the days as profiling through the days. Now, the last little piece I wanted to share quickly was some research on oligodendrocyte protection. So here I was kind of using what I learned in just radiation biodosimetry and also neurodegeneration to marry the research, like using metabolomics, central nervous system damage, and radiation injury. Here I was looking at the actual metabolism associated with radiation injury and demyelination and testing out a potential drug therapeutic. This is just showing the effects of demyelination. This is in a mouse after exposure to radiation, and in particularly in the corpus callosum. This is an area of white matter that is associated with demyelination and associated with that cell I talked about, the oligodendrocyte. Again, we're able to do whole brain metabolomics just to be able to distinguish some of the metabolism that was associated with this change in injury from radiation. And we picked out the TCA metabolism. This is actually involved very centrally to the cell. And if you teach biochem like me, then we talk about it all the time, tricarbolic acid metabolism. So we tested out a particular drug that I had worked with previously in neurodegeneration that targets this metabolic pathway called dimethyl fumarate, and we wanted to see how this would affect radiation damage in those oligodendrocyte cells, because we knew that it could help with oxidative protection from past research that I had done, because radiation damage and oxidation damage are very similar and they have a lot of the similar pathways. We thought this might be very helpful. And this is just some of the work in the cells that I did, looking at changes to proliferation. And you can see there's also some changes in the senescence of the cell if they were either treated with that drug via not being treated with the drug and then exposed to radiation. So they proliferated better. They had less senescence. We actually looked at their ability to produce myelin and lipids normally as well. And again, the addition of this drug DMF seemed to encourage more myelin production even after exposure to radiation. We also did a small pilot in mice as well. And again, looking at that corpus callosum region to see how that was changing when we were using this drug protection via no protection in radiation exposure. And you can see the myelin production. This is a stain of myelin down here. That is marking the neurodegeneration was not as marked in the DMF treated mice. So there's my acknowledgements. I did a lot of work in the radiation with Georgetown University and Dr. Fornace's lab, who has kind of pioneered some of the work with the DoD, as mentioned, in using metabolomics for biodosimetry there. And then I kind of offshooted using my neurodegeneration and tied it all together in my postdoctoral work. And one last shout out, I actually am from Cape Canaveral, Florida, and I grew up with NASA in the Space Center. Both my parents worked on the space shuttle projects in different capacities. So I am just excited to be able to talk to you, those of you from NASA in general. All right, that's it for me.
Okay, thank you. Can I ask some questions? Or?
Of course, yeah. I tried to keep it very brief and went through fast.
I just don't know what Dr. Xu's plan is for everybody to complete the presentation, then we will discuss, or? Okay, then if he didn't say anything, let me ask some questions then. Oh, of course, I see some familiar names in your last slides, like Alphonse. I mean, he has some of the NASA grants, and Zhen Hauser, is that at Arizona? Yeah. Okay, I think he once visited us here at JSC. And yes, so I kind of, from what you presented, you have some projects from NIAID or to do this
yeah,
kind
when
of.
I was a postdoc, I worked directly with NIAID. I worked, as I showed, in Dr. Fornace's lab, paired with, as you noted, Arizona State. That's or Arizona University, where I worked with that polymeric kind of project where we were looking at that, and also up in Colombia as well. So there's ties there for NIAID.
I think he had he has some project from Colombia too, I believe. Yes, that's great, and I think that certainly addresses some of the NASA questions, and especially, yeah, I didn't when you said your field is metabolomics, and I didn't know. I mean, it was related to brain CNS damage, and I thought it was the liver metabolism. And so I said earlier that of course that's one of our concerns, and of course, I mean, in all the CNS is one of the major risks, and if the type of studies that you have done definitely will have some association with the CNS risk that we are concerned about. And with CNS, it's not just the radiation. Yes, I mean, it's radiation. I do understand that the type of dose will be usually high for our interest. I didn't get into so much details, but for space, and normally you will get, let me use the gray then as a unit. You probably get maybe close to 1 gray for a mission to Mars for 3 some years. And then you probably can get a whole body dose of 1 gray, perhaps 2, I mean, during a large solar particle event. So we usually won't reach to the level of 4, 5, 8 grays that the Homeland Security is interested in. So that's, but then nevertheless, I mean, the damage is different, because I always think of the gray being a physical quantity. I mean, it's the average of over volume, but then that's with gamma rays. But then with charged particles or space radiation, you can deliver very high dose or high level of damage locally, which can be similar to high doses of gamma rays or low LET radiation. Yeah, so I think yeah, so I would say what you do definitely would be interested to the CNS risk. Now, of course, I mean, another area of CNS is not necessarily a radiation, it's microgravity. And I didn't mention much about microgravity, and actually in recent years, my my research has focused more on the combined effect of microgravity and radiation exposure, and so how will microgravity affect those metabolic activities, and how that will affect the DNA damage response or DNA damage repair. I mean, those are all the questions that we ask.
Yeah, I'd love to learn more about how to work on projects with NASA doing these types of radiation experiments that are, like you said, more tuned toward what you're going to see in spaceflight, as opposed to things that high gray, intense, or X-ray, gamma that we're not really concerned about. We're more concerned about for defense reasons, but not for NASA.
Yeah. As I said earlier, I don't know how much this metabolomics information can be used for biodosimetry in the blood, because you cannot take the brain tissues, right? So in the blood, and from the data that we have collected, although we haven't published the data, but actually we try to focus on identifying anything that's related to metabolic activities, but we didn't quite see it.
Yeah, I think I've been using it more towards finding biomarkers that are of interest related to radiation that we can then target, and not necessarily, like you said, biodosimetry at these low levels, because especially for brain too, and at low level radiation, trying to use the blood is very hard to find markers that are going to be useful for biodosimetry reasons.
Great. That was awesome discussion there. Again, that's not my field, so I will not try to be expert on that. So I guess that's wonderful. So is there any other faculty member who ready to share your presentation? Please go ahead. Dr. Uduchu and also Dr. Lee, open your camera. So please feel free, either one of you, please go ahead.
So I have to upload file myself, or you upload?
You can directly share from the screen. There is a like a button on the near the microphone. and you click that one, then you will be able to share your window or your screen. So then you can.
Dr. Zhu, one quick question. Are we ending this at 12:30?
Yeah, thank you for reminding us. We're running a little short on time, but I you know, I want to respect everybody's time, but also I want to, if, you know, Dr. Wu is available, we probably can like stretch a little bit, if everybody is fine with that.
I'm okay.
Okay. Thank you. And if anyone of you have to leave early, you would like to present early, yeah, please let me know. Yeah, and so Dr. Lee, is this what your slide?
Yeah. Can you see it?
Yeah, I can see it. So
Okay.
I would yeah, I would just say maybe for the interest of time that
Yeah.
if whoever want to present, if you can just yeah, limit it to 10 minutes. So
Okay, so I only need 5 minutes.
Okay, perfect.
Okay, this is Dr. Lee, and Dr. Song is the leader. So we have been doing some interesting chemistry. So this one, the title is called uranium extraction from seawater. We work with University of Maryland and Department of Nuclear Engineering. We made polymers and fabric and functionalized with organic ligand. So those materials can successfully extract uranium from ocean. That's one of the projects. So we published some paper in this journal, AST. And so the chemical, this is another one. The chemical we use, the ligand is here. I think at the very end. So this kind of ligand, we have OH group and sulfur and nitrogen double bond. So the metal of uranium will be in the middle. So they can very effectively extract that. So that's one project we did. And the second one I mentioned in the introduction, we have a seed grant from DC Water Resources Center. We put some interesting organic chemical or ligand on the polymer. It's called bromo-1 resin. We put on the resin, which is a stable free radical. And the free radical can be reduced by using vitamin C or some other reagent to change into a strong reducing agent. And reducing agent can be used to remove the chlorine. Result here, very qualitative. So the vial 1 is the control. The vial 2 is the tap water we simulated. We're adding some chlorine in the vial 2. So if treated with our material, the resin, the chlorine can be removed successfully. So that's the C grant. So to show us the material we use can be remove oxygen. And the reason why we propose this kind of organic ligand into the polymer for water treatment is the small molecule I put here. When I was working for a pharmaceutical company, a very similar organic compound we synthesized and submit for FDA for phase 1 and phase 2 clinical trial for eye drop. The eye drop, the purpose of eye drop is try to reduce the cataract or some kind of oxidative damage to the lens. And we were very successful to make the eye drop and got a patent. And we did a phase 1 and phase 2 clinical trial. And at the end, the eye drop formulation was sold to some other pharmaceutical company. So I think, as Dr. Wu mentioned, in the space, in the universe, when the astronaut go to the sky, may cause some damage. I think that this kind of compound, we can modify and make them patentable and to check the protection effect from radiation or from some other damage. So that's one area I think we can pick up quickly. And the second, I'd like to mention, Dr. Song and our group is very strong in organic chemistry, organic synthesis. So we can make different organic compound for your research group, for NASA, and to serve different interests. I think that's all I like to talk. Thank you.
Thank you very much, Dr. Lee.
Okay, thank you. Yes, and I have to say, I mean, this is probably not my area, but then I do want to say this. Of course, development of countermeasures that mitigate radiation risk, and that's the one of the main focus areas within NASA. And so anything that that has the antioxidant properties normally will have some radio protection effect. So certainly, I mean, especially when such a compound has a low toxicity, and definitely we are open to look into those. So yes, that would be one of the applications. And the other, again, I don't quite understand, that's not my area, but they're extracting, well, in vitro utilization of the resources on the moon or Mars, and for long-term survival, and definitely, I mean, that's an interest of NASA. And I can give you an example, and then you can kind of think of something that your research might fit NASA's needs. And the example is that one professor has a project before that they just use some polymer materials, just a glue that they glue the lunar or Martian soil together to make the bricks, and then you can use those bricks to build a house or build some habitats on the moon or on Mars, and that kind of technology will be of need for NASA in the future too. And then, of course, if you can extract water from the ice, and you can extract some soil, so you can grow plants on the moon. Just a general statement.
Okay, thank you very much. Yeah.
Thank you.
Thank you, Dr. Wu. So Dr. Lee, just for your information, and Dr. Song, I saw a recent announcement from NASA. Actually, I think it's relevant to that area. So I'll share with you, see whether you will be interested in pursuing that. So.
Okay, sure. Thank you.
Mm-hmm. So so I guess next we can have Dr. Azam. I know you have to leave, so if you want to share your slides. And then we'll have a couple other will be sharing their research as well.
Thank you, Dr. Zhu. I appreciate this. Actually, I want to start with the exciting news we just heard. So one of my proposal with my student, like with the NASA MSI fellowship, got funded yesterday. So very exciting to have NASA funding this. And we are going to look into ethylene scavenger, like different materials, to look into sustainable, aquaponic system for space exploration. So thank you, Dr. Zhu, for bringing that opportunity to me, and your encouragement is really helped to achieve that. So as the time is limited, I want to go quickly. Dr. Wu, your research area is slightly different than what I do normally, but I think there could be scope that could be overlapping. So I will go a little quickly because of the time. So as you are very familiar with it, but all the colleague also knows this, they have different risk types they look into. Of course, you are looking into this type 1 risk on the radiation effects. And when I looked into the type 2 type of aspects that NASA is looking for the advanced life support, all of the aspect is actually where I can contribute as a researcher from UDC. And I will highlight some of the projects soon on the side of advanced life support and on the food and nutrition and environmental health. As you know, Dr. Wu and others, that they have their advanced water recovery system in this in the ISS and in the space exploration. They have the urine processor assembly, and then they have the water processor assembly, and then they have oxygen generation system, and they have a CO2 reduction system. I have some ideas of how I can contribute to this type of aspect of goals NASA has, especially for long duration of space exploration like Mars and others. As I told you, Dr. Yu, when I was introducing myself, my main area of focus in water wastewater treatment, looking into carbon removal and absorption, nutrient removal and recovery, and sludge treatment and energy recovery, looking into also some algae aspect of it. And I look both process chemistry, like advanced instrumentation tools, and also like microbial ecology and advanced molecular tools to look into different aspects of water wastewater treatment, water use, biocorrosion, and things like that. So this is one of the projects that actually we have unofficial oral confirmation that it might get funded. So we are not from NASA, but from our DCWRI grant. So looking into next generation modeling techniques for determining nitrogen and phosphorus from point and non-point sources for harmful algal blooms in the US. And I know NASA has some different areas of land and vegetation, ocean climate type of aspect that can be a good fit, like a remote sensing data that we can utilize for this particular project. I have another project that could be and this is the project I was mentioning that just got funded by NASA yesterday, that aquaponics with ethylene scavengers to support human life of Earth. So we are looking into a different type of recoverable in situ materials in the space or in Mars that can provide some synergistic roles in water treatment, air quality, food production. So you can see the diagram here: food production, waste treatment, water recovery, and air management. And we like to use some materials for this.
I don't see any. Did you share anything? Because I don't see anything though. I only see Dr. Shi's.
Oh.
I don't know whether you have been sharing anything. So, okay, now I see it.
Okay. So I was talking briefly. Then this is the advanced, life support system I was talking about, the food, nutrition, and environmental health, where I could be providing a very strong support. And I was talking about this type of water recovery, water processes assembly, urine processor assembly, and oxygen generation assembly, and CO2 reduction system. I can provide a specific guidelines into each of the processes. And I was telling you briefly, I'm going to skip that because I sorry that I didn't know that you are not able to see. But I focus on a lot of carbon removal, nutrient recovery, sludge treatment for water, wastewater, looking into chemistry and microbiology, and for different water use reapplications. But I was talking about specific projects like this. I have unofficial confirmation that it will get funded. We are looking into some remote sensing data to look into harmful algal bloom. I know NASA has some program that funds this type of projects. and I will love to collaborate. Anyone you might know, Dr. Wu, who might be interested to fund this type of projects. The second project I was talking about is related to different type of recoverable in situ materials. So this is the project B that just got funded yesterday, Dr. Wu, from NASA. Aquaponics with ethylene scavenger to support human life of Earth. Looking into different type of materials that we can recover and to enhance air quality, water recovery, and waste treatment. And then some other projects could be very interesting to look into. I know you guys have a waste problem also. Some of the organic waste that you generate might be, we might be able to convert them to some byproduct that we can utilize in the food production system or water quality control or air quality control, and maybe generate some energy like hydrogen in the system. I have some existing project that I'm working on. And urine can recover struvite, and there might be some corrosion problem in your airspace. So I have a project that I'm looking into struvite dissolution and inhibition with recovery, and we can use struvite as a fertilizer. We can use it for absorption purposes of some of the pollutants, air and water pollutants that we might encounter in the space. So those are the kind of basic projects that I have. I would say directly relevant project is that funded immediately with from NASA, but we can collaborate in other areas. I don't want to take more time as we are limited in time. I just want to end this, Dr. Wu, and if you see, and all these pollutants might have some effect. You look into radiation, but all these pollutants also have similar effect on cancer causing effect and carcinogenic effect and DNA damage effects and things like that. So that if I'm not sure you will be able to look into that aspect of it, but there might be some overlap there with Dr. Tarabolity and what you do.
Yeah, thank you. And first, congratulations on your NASA grant. But first question is that from which center or which program did you receive the funding?
This is called NASA MSI Fellowship. Dr. Zhu, do you know specifically? The signature came from manager and NASA Fellowship and International Initiatives, Mrs. Caroline Nolens.
Okay. All right. Okay, but yes, of course. I mean, now apparently, I mean, you have done a lot of research on what NASA's interests are, and yes, I mean, my area is radiation effects, and of course, I mean, we are concerned about cancer and other health consequences from exposure to toxins in space. And then the water quality, air quality control group is within my branch. And so this recycle, and so they do this type of detection, monitoring on the ISS to make sure that the astronauts are in a safe environment. And but then within the habitability, that's another division within JSC. And I certainly, I mean, can introduce you to some of the folks both within my own branch and in another division.
I appreciate it, Dr. Wu. Dr. Zhu, if we can follow up about this and follow up with Dr. Wu and make some contacts with his division and other division, that will be awesome.
Yeah, definitely. I'll share Dr. Wu's contact information through the chat, so please feel free to reach out to him directly. Yeah.
Okay. Thank you.
Thank you, and congratulations, by the way.
Thank you.
So I think we have Dr. Udutu. Yes, please go ahead. Yeah.
Thank you. Nice to meet you all again, and thank you for this wonderful opportunity to collaborate. Basically, I won't take too much time. I just want to give you a kind of overview of what the work I do. Okay, let me do screen share. I don't know if you can see my slide. Is my slide evident?
Not yet.
Let me try this again. Oh, let's see. Am I doing something wrong? Okay, let me try this again. Sorry about this. Let's see. Oh, well. Okay, I'm sorry. Let me try this again. No. Okay. Research presentation. Okay, try it. I want to try it one more time. Is it showing up now? I think it should be up.
Not yet.
I have your slide. If you need,
It's coming up. It's coming.
Okay, come up.
Is it up now?
Yeah,
Yeah.
great.
Okay. I will just I'll just pick the very important relevant slides here. So here I just want basic information here is to show the utility of spectro-electrochemical methods for investigating structural activity properties of electroactive materials. And so, like I said earlier, I'm associate professor of analytical chemistry, and my work actually focuses on spectroscopy, electrochemistry, material research. I do a little bit of synthesis, modification of functional groups, especially when needs be to achieve my research goals. Here, just some of the current trends. Some of the current trends in the area of spectral electrochemistry. You see here, normally I use, like I said, spectroscopy and electrochemistry. But when we say electrochemistry, normally I employ cyclic voltammetry, where I'm looking for kinetic information and reduction potentials of materials on the surface or in solution. I also employ UV spectroscopy to examine electronic properties of an aggregation state of important compounds. And at times I combine potential, of course, UVVs and electrochemistry. When I combine UVVs and electrochemistry, I'm examining potential dependent behavior or potential dependent UV spectroscopy of properties of compounds. And then we introduce Raman spectroscopy and surface enhanced Raman spectroscopy, which provides us with molecular information of adsorbates. So below you see the sort of work I do. Basically, we are trying to, one area of interest I'm looking at now is the single entity electrochemistry. Basically trying to understand the behavior of single entity molecules, nanomaterials, the single entity of those on surfaces. And this is very important for nano impact studies, and as well as important for particle size determination. We're trying to see there how particle size influences properties of nanomaterials, as well as examining electronic structures of compound. So here for this, basically for this specific case study, which I did in the past, is already published. We were examining pH-dependent aggregation state of adsorbed ion tap. And when we say ion tap is basically a porphyrin, ion porphyrin, which is similar to chlorophyll in plants. And ion tap has, of course, it's a redox enzyme. It's capable of energy conversion. It's capable of transforming energy, of course, from one form to another. And for fuel cell, basically capable of catalyzing the reduction of compounds, serving as catalysts in fuel cells. And that was what we're looking at as far as this study was concerned. Fuel cell technology is one technology that is very, very important for NASA. I know NASA is working on hydrogen fuel cells, hydrogen fuel cells, and so on and so forth. So it's a very important study as far as that's concerned. Fuel cells, we know, are devices that convert chemical energy to electrical energy. But this time around, the focus in this work was to see how oxygen, which is very abundant, can be used in the cathodes of fuel cells. If the choice of material has to be a material that's readily available, it has to be if we can generate water as a product, which is harmless, that will be a very great breakthrough. However, in the area of fuel cell technology, one limitation is usually the slow electron transfer kinetics. And so as a result of that, you have fuel cells that have limited power densities, don't produce enough energy. And so the solution, in a nutshell, is to have a material like porphyrin, which we are investigating here, that can catalyze, speed up the electron transfer kinetics. And that's why we chose a material that's similar to chlorophyll. That's iron tap. And chlorophyll has magnesium, but iron tap has iron at the center of the complex. The iron tap was synthesized, of course, for this purpose. Here is just to show you a little bit of mechanism of oxygen reduction. Oxygen, this is the mechanism. And so this is a well-published, of course, established data that oxygen can be reduced, but slowly. And so in this work, our goal, just as other researchers have seen, that cobalt, manganese, iron porphyrins, general iron porphyrins, not iron tap, but just general iron porphyrins can reduce oxygen. And so what they will do is polish electrode surfaces with, or coat electrode surfaces with these porphyrins, and then see how they catalyze the reduction of oxygen. And in those studies, they identified that the mu-oxo bridge iron porphyrin dimer is a very active form, is a very catalytic form of iron porphyrin. So in our study, which in a nutshell, we propose that this novel compound, iron tap, will, of course, be able to reduce, of course, to catalyze the reduction of oxygen on electrode surface. And so that's why we needed electrochemistry, we needed UVV study, potential dependent UVV study, we needed CS, surface enhanced Raman spectroscopy, and Raman spectroscopy to do this work. And the real intellectual challenge here is whatever material of choice that should be used should be chemically stable. It should be able to absorb on the electrode surface. It should be reversibly, of course, changed, oxidized, reduced reversibly. And so that was what we set out to do in this work. And we're testing iron tap. And time will not permit me to go into the details. It's been a published work. We went into extensive study with UVVs. We were trying to find out, can iron tap, since the dimer is responsible for catalytic reduction of oxygen, can iron tap, does it have the dimer form? So we're looking at this. We saw there was a different, the monomer form of iron tap at pH 3. We found the dimer form of iron tap at pH 11. So that was one breakthrough there. And then I went ahead to fabricate. Actually, I fabricated this device, the OTTLE cell, optically transparent thin layer electrochemical cell. This cell allows us to, to examine the spectroscopic property while, you're changing the potential of the surface of the cell. And so we're able to generate very important results. I know we're short of time, and I wouldn't really want to bore us with the details of this, but we're able to demonstrate that iron tap was in the monomer form at pH 3 and in the dimer form at pH 11. And we did chemical reduction at pH 3 to back up with the electrochemical reduction. We also did the chemical reduction at pH 11, and then we proceeded with CS, and which is surface enhanced Raman spectroscopy, which looks at the surface of electrodes to see whatever is happening in solution, can it also be demonstrated? Can ion tap adsorb to the surface? Because in the fuel cell, you need the catalyst to stay on the cathode, of course, to be adsorbed on the cathode and be stable on the cathode. And so that was what we went in here. We were able to, of course, discover that ion tap we identified the bands that were responsible for the adsorption and bands that were responsible for the binding of ion with oxygen on the surface at pH 11. And of course, we worked with reference to literature, and we were able to characterize that ion tap was existent actually at different pHs on the surface. And I want to end by just, I know we don't have time. Basically, our conclusions in this work was showing that ion tap exists as a monomer at pH 3, as a dimer at pH 11, and it's the dimer that is actually responsible for the catalytic reduction of oxygen. And I end by saying that currently our future studies we're looking at exploring the electrochromic properties of light-activated nanocrystal quantum dots particles, because I'm trying to harmonize, my group is trying to harmonize or use artificial intelligence to develop predictive models for structure-activity relationship of quantum dots, zinc oxide. And we're hoping that we'll have some breakthrough, and hopefully NASA will be interested in some of these studies. Thank you very much for your time.
Thank you. Yes, of course, I mean, this is not quite my area, but then you already said that the fuel cell, of course, will be an interest of NASA. But one application, I mean, this is the radiation dosimetry that I mentioned earlier briefly. So again, I mean, I probably will use this my colleague's project as an example, and then you decide how your research can or cannot be applied to this. And that's the development of a radiation dosimeter. So basically, my colleague's area is not radiation. That's how I support them with the radiation exposure, with some radiation background. But then he's a nano guy. He has some nano materials. But then what happened is that you have some materials. It doesn't matter. It's a nano, or I think in your case you have some device, and the radiation exposure will break the bond or create some radicals, will affect the electrical properties. And if this effect is significant enough, and for instance, if you put the electrodes and then radiation exposure will affect the current, and then you have a radiation detector. So I kind of, from what you explained in the beginning, perhaps, I mean, your device might worthwhile just to explore that kind of possibility.
Okay. Thank you, Dr. Wood, and thanks for the idea. I will certainly look into that bridging point of radiation detectors. Thank you very much. Okay. Let me stop sharing.
So I think we have Dr. Delac will present next. I know Dr. Sneed also expressed interest in presenting. I don't know she's she's still interested in presenting. So maybe after Dr. Delac, we'll see whether she will like to present her research. One more thing I forgot to kind of mention earlier is that we had let me see whether Dr. Dixon, I don't know where she is still with us. She had a little microphone issue at the beginning, so we couldn't introduce her. So I just see whether she's, let me see. Seems like she has left. So anyway, so she is a professor in the biology department, I believe. Yeah, so she is also heading one of the NSF center here. So yeah, so I would, if she joins us back later, I will request her to kind of introduce herself. So Dr. Dilek, yeah, please go ahead. I don't want to take your time, so please go ahead and share your research.
Well, I like the title already. Live cell images. I think you're muted. You are muted.
Oh, sorry. Okay. So I cannot able to make it like a full screen, I think, right? Is it, can you able to see full screen here? No, right?
Yes, yeah, I can see it clearly.
Yeah, I think it should be fine. Yeah, we can see that. We can see your screen. Yeah.
I cannot even see what I'm presenting, so that's weird. Okay.
Oh.
And that's my part. Let me see if I can show the screen. Just a second. I will actually. Okay. Can I go this direction? Can I go this direction? Yeah, I think it is fine. Okay, yeah, Dr. Wood, thank you for your nice presentation, and also I would like to thank to Dr. Zhu for making this event possible for us. So my part is a little bit different because I'm not working on the radiation, obviously. I mean, I have lifestyle imaging applications. I am in a different part of the chemistry. I am in the chemical biology. I'm a chemist, but I am doing chemical biology. So our work is developing new chemical probes, mainly fluorescent probes. And we are also developing new bioconjugation tools for live cell imaging. So our lab is basically designing small molecules like probes for the appropriate application. We are designing like also these molecules like kind of organic synthesis. We are doing organic synthesis of probes, and then we are checking their spectroscopy, whether they are actually applicable for the live cell imaging. It is basically what we are doing is UV visible kinetics applications, and we are doing also mainly fluorescence applications. And after we confirm these probes are actually useful to be used for the live cell, then we are actually going forward for the next application. So these recent projects that we are having right now is one of them is the oxidative stress induced carbonation. That I'm just catching up from your presentation is just like maybe the NASA might be interested in like technology development of like small molecule probes to detect the any type of oxidative stress based applications. But of course, I'm not sure about it. So our part is we are designing different library of fluorescent probes to detect oxidative stress induced carbonylation, which is actually relevant to the cancer. So the reactive oxygen species, which is causing oxidative stress damage and which can trigger the cancer and metabolic diseases. So our idea was that is it possible to detect or monitor this carbonylation level. And we focus on the, of course, in this project, mainly to the carbonylation process. So we were normally focusing on different type of probes, like we were synthesizing body piece, coumarins, and all kinds of other dye base molecules. But for this project, by coincidence, we actually ended up with this molecule, small molecule. And this was not supposed to be fluorescent, but it ended up like a fluorescent molecule when we started working on. So it's kind of a hydrazine-hydrazone formation like we are seeing here. What is this meaning is in organic synthesis, it is meaning something, but in biology, we are making a molecule in the middle, which is not fluorescent, but with the functional group of the like amine derivative, which is hydrazine, when it is reacting with the any aldehyde groups or carbonyl moieties inside of the species, it is turning on, which is giving light to the outside. So this kind of a commercial available DMPH is used for detecting the carbonylation. And we are actually develop a kind of a similar molecule, but not the same molecule, which is detecting the same type of aldehydes or ketones or any carbonyl species. We also developed some kind of a molecule which has a boron inside. We patented that project, I mean that part of the project, and then we licensed it to one of the companies, Click Chemistry Tools companies recently, so that is commercialized already. So we are in the technology development of this part. So we actually developed this, and then we decided, okay, so can we able to use this in the live cell imaging? Because this is a kind of a small molecule, but it is not very, doesn't seem to be very soluble in water. And we try to check their spectroscopic properties first to make sure that they are actually giving really good results for us. So absorption emission values in the range-wise, it is in the 300 or 400 ranges, but emission maximum is actually in the 460s and 500 regions, which is, we are seeing a huge shift on the absorption maxima. And fluorescence-wise, as you can see on the pictures, we are seeing turn off and turn on when we actually react this probe with any carbonyl species, mainly aldehydes. We check with the model aldehyde groups in our lab. It is turning on. So the question is that we are asking is it possible this molecule to try in the live cells? So we tried renal cancer cell line. We had renal cancer cell lines in our lab, and we have HDF as a normal cell line, fibroblasts. A498, ACHN cells, the last two categories are the renal cancer cell lines. One of them is metastatic, and we apply hydrogen peroxide to the cells to make it more oxidative stress environment into the cells. And after that, we are adding our probe. This is the synthesized probe. And then we are actually changing the conditions of the cells like 10% FPS and serum start. So what we have seen is when we start the cells, serum start cells, we have seen a fluorescence coming up. It is turning on the cells, which is interesting because in the HDF, we have seen this, but we have seen A498 and ACHN a little bit more, which we don't see this fluorescence turning on in the FPS one. So this gave us an idea about that it is possible this can be explained with the CMAT mutation, which is the metastatic potential ability in the cancer cell lines that is totally activating that system. So because we are actually, when we are doing the serum star cells, we are actually generating some kind of extra reactive oxygen species, which is actually triggering the cell death. So it looks like if we were observing that turning on a system which is triggering oxidative stress, which we are thinking also that is actually making more carbonyl species into the environment oxidative stress wise. So we published this one like recently. So then we checked the same results in the presence and absence of pyruvate conditions, because pyruvate is also kind of a weird molecule, which is actually blocking the fluorescence, which we found that thing just recently that it is kind of the pyruvate is like when it is coming into the reaction, it is reacting with the hydrogen peroxide. And when we have the probe inside, it's possible that our probe is reacting with the pyruvate in the media. So we remove the pyruvate in one cell line, and actually we have seen it, the fluorescence is coming up again backwards. So the condition, we play with the conditions for a while. And then the second project that we are working on as a tool developing, this is a new project. We just recently get the preliminary data. We didn't publish anything else. Photocatalysis strategies. So we are using a light to do the chemical probes. So this is a kind of organic synthesis steps. Instead of using a conventional method, heating up, doing normal reactions in the chemistry in a flask, we are actually doing the same reactions, doing the substitutions on the benzophenone molecule, but we are using a visible light. So this reaction actually working, and we started this project in NIH. So I was working at NIH last 2 years in summer as a visiting scientist. So they opened their lab for me, and then we started the project. So we got the preliminary data, and they are collaborating with us on this project. So this is a kind of a unique photocatalysis approach, and our idea is here to, again, technology development wise, to use this light technology to do the product or some kind of a cross-linking agents for the synthesis or some pharmaceutical applications. This is a kind of phototoxicity wise also it will affect, because the toxic environment that we don't use it for doing this reaction, we are using just the normal light to do this reaction. Basically, that's all for my project. So we are right now recently collaborating with the Georgetown University Medical School. So they will try to do our probes to use them in their tissue culture. So Dr. Levy, we are collaborating with them. I'm also recently joined to Georgetown Medical School as an adjunct research faculty. So we will continue this collaboration with them. NIH Chemistry Synthesis Center is the one that I work with. So photocatalysis project we developed there. So they are the other pictures that you are seeing, all my grad students and undergrad students before. So I'm trying to put new students at UDC, so upgrade the lab and then get started on these two projects. Thank you.
Right. Thank you. And I said earlier, when I see the word live cell imaging, I thought, gee, great. Yeah, of course, live cell imaging is great. And for two reasons. One is, of course, science. And science is that the NASA has this space biology program, and which addresses those questions like how life can adapt to this space environment or survive in the space environment. So then so they will study microorganisms, plants, and other species. We are more human. So I think any processes that will occur, I mean, in the space environment, from exposure to microgravity to space radiation, and then that will be the interest of this space biology program. And two, of course, is the technology. I mean, any technology that can allow live cell imaging, that's always very interesting. And I know that there are projects. Actually, my colleague has a project to develop microscopes, simple microscopes on the ISS. So if you're live cell imaging, I mean, you can make it small light. I mean, those are that would be a technology development area. And I probably should mention about the NASA SBIR program, which most of the government agencies have. And so some of those, if it falls into the technology development, then there might be opportunities to get some funding from this type of programs. And normally for phase one, which is not a whole lot of money, because I'm a subtopic manager for the radiation subtopic, so I know a little bit. And then you get $125,000. I mean, just it's for proof of principles, and if you can develop this kind of technologies for space, space applications, I mean, you might be able to pursue that kind of a direction. And of course, SBIR, you have to start with start a company, and which has been done by many of the university faculty members.
Thank you so much.
Yeah, thank you. I see Dr. Sneed. I don't know, Dr. Sneed, are you planning to present your share your research?
No, I hadn't planned on presenting today. I was very interested in attending just to see essentially how could planarians fit into, I guess, a NASA mission, but how relevant could they be? So really trying to learn more about how I could actually contribute to this. So thank you for inviting me. Yes.
Yeah, sure, no problem. Thank you. Okay. Yeah, so I know we are way over our planned time, but I really appreciate everybody for staying and presenting your research and just participating and sharing your interest. And also, of course, Dr. Wu for staying with us and listen to our presentations and provide your feedback. Just, you know, I mentioned earlier that this is just the beginning to start a dialogue. So if you see any opportunities, you can, you know, have research support from NASA. You know, we're here to support and to connect you. And also we have, you know, the OSTEM office at different NASA center is also there to help us with connecting with the subject matter expert. So definitely we look forward to have more conversation on different areas in the near future. So I don't know anyone have any questions or comments at this point, but I would just want to thank everybody for for spending your time with us this morning. Yeah.
Okay, thank you. And certainly I encourage everybody to contact me.
Yeah, definitely we will. We'll send a lot of emails to your way.
Okay, great. All right, thank you very much.
Thank you, my colleagues. So we have another one in the afternoon. So yeah, if you're interested in attending, yeah, please let us know. Yeah. Thank you. Bye bye.
Okay, thank you.
Okay, thank you very much, Dr. Wu, Dr. Xu.